Hello everyone. Today I'm going to talk about my research work, which is exploratory analysis of orphan genes using unsupervised learning on single cell RNA sequencing data. My name is Harsha and I'm a second year master's student in computer science department. So this is a brief overview of my research work. So single cell RNA sequencing analysis basically analyzes gene expression data at cellular level, and this is a new methodology of studying gene expression data. It's useful to study complex organ development and cell types. So uh, previously this was not the case because there was no fine-grained data at cellular level. And computationally, this poses a lot of challenges. The first thing is the data sizes are large and the data is itself very sparse. That means most of the cells do not express a gene, so it will be a zero. And we are planning to create a enhanced pipeline for single cell RNA sequencing, which will be used to analyze orphan genes in humans. So this is a workflow of single cell RNA sequencing, typical workflow. The first one is expression quantification. This is the first step, and this involves alignment of the reads, the reads of the expression to a reference genome. So, for example, if we have collected tissue from human brain or human kidney, so we will be aligning the reads obtained from from these cells to a human genome, and once we align that, we obtain this quantification, this uh, the expression matrix, and this will be used for further analysis and unsupervised learning. So once we obtain the matrix, we pre-process the data. We normalize and scale the data. This is a regular statistical process in order to perform principal component analysis and other dimensionality reduction techniques. And once the data is pre-processed, we perform the computational analysis. Uh, so this could be linear dimensionality reduction, k-means clustering, or we could have a t-SNE or UMAP. Those are nonlinear dimensionality reductions. So in a biological perspective, we initially have a, a group of cells which are extracted from a tissue, and these are initially not segregated into cell types, and we do not have a way to identify cell types and which cell belongs to which cell type. So once we perform this analysis, we can identify cell types, and we can identify which cell belongs to which cell type. And this entire process is unsupervised, so we do not have any labeled data. And the problem we are trying to solve, uh, basically there are multiple problems, and the first one is performing unsupervised learning on this high dimensional unlabeled expression data. So typical sizes are 60,000 genes and 30,000 cells. So the matrix is quite large, and so it suffers from high dimensionality, and we cannot visualize such high dimensional data. And performing computation on such high dimensional data requires resources as well. And the data also suffers from a sparsity problem, as we've seen earlier. So most of the matrix is zeros, um, and so it requires some cleaning and preprocessing before we can analyze it. And we also want to implement this this single cell RNA sequencing pipeline into a scientific software package. And this is an open problem. And then another problem is identification of cell types with uh, high orphan gene expression. So we are analyzing different tissues to identify highly expressed orphan genes. Yeah, and there is a lot of unexplored data in this field. And the solutions we are working on are first thing is using linear and nonlinear dimensionality reduction techniques such as PCA, TSNE, and UMAP, and to optimize the algorithms and existing workflows. So this will address the high dimensionality problem and uh, high resources problem. And then we also are looking at usage of statistically sound scaling and normalization steps. So this is a prerequisite for the dimensionality reduction. And this will address the sparsity problem. And then we are also working towards implementing this pipeline and incorporating it into MetaOmgraph, which is software developed by the research group which I'm working in. It's Dr. Vajjhala's research group. So this will address the lack of tools. And the next solution is to create a cell atlas to find interesting orphan gene expressions. So cell atlas is basically the output of the unsupervised learning. And the limitations of the current research work are, the first thing is the normalization and scaling. They do not eliminate sparsity. It reduces it, but does not eliminate it. And another limitation is the pseudo alignment tools, which we are experimenting on. These are computationally very efficient, but they have low mapping rates compared to an actual alignment tool. And then another limitation is in the software engineering perspective. So implementing this pipeline, this analysis pipeline in a desktop software is not quite feasible because of the resource constraints. And yeah, so right now there are not many tools which can perform this alignment, this analysis. And the future work would be first thing to remove biological and technical variability. So this poses problems in the downstream analysis, and which could result in some incorrect statistical analysis. So researchers are working on this problem using probabilistic generative models, and in order to capture low dimensional representation of the data. And also people are working on software packages that can utilize the remote servers for computational to obtain a good computational advantage. And the related work to this research is there are a lot of related works, but these are a few of the important ones. The first one is a paper on UMAP, which is a really popular technique to visualize single cell, to reduce the dimensionality of a single cell data and visualize it. And the second one is a paper which compares different technologies and the studies on different species. And this lab actually created a R package called SURAT, which is quite popular for single cell analysis. And the third paper is a single cell analysis of human brain tissue. And this is a good reference for the orphan genes exploratory analysis, which which we are currently doing. So that's all I have for my presentation. I hope you liked it, and so if you have any questions, I would be happy to take them. And this is my contact information. Um, Thank you so much for listening to me. See you.